Good evening, everyone. Am I audible? Please confirm. And is the slide also visible to you all? Am I audible? Please confirm. Okay. So today we are going to discuss about Unit 1, important questions from here. Here is the first question from December 2016. This is regarding Ramachandran plot. The following are four statements on peptide protein conformation. Glycine has the largest area of conformationally allowed space in the Ramachandran plot of phi and psi. Okay, you have to answer which are the correct statements over here. And B is saying a 20 residue peptide which is acetylated at N terminus and amidated at C terminus. So this part you can ignore as of now, because this is just the N terminal modifications which are done in the peptide so that the folding will be happening properly. And what do you mean by acetylation? You have to check once whether acetylation is, generally you do acetylation at N terminus only and amidation at C terminus. So this you check, and then you can ignore. So a 20 residue peptide which has phi and psi both in minus, as you can see minus. This is plus minus 5 degree for all the residues. Hence, it can be concluded that conformation of the peptide is helix turn strand. Helix turn strand is a motif. Motif in proteins are a group of secondary structures which has a certain kind of stability. So you will have the helix, and then you will have the turn, okay, and then you will have the strand. So this is what you have. The allowed values for phi and psi are for amino acids in a protein are not valid for short peptides. And a peptide which has, so again you are seeing these are the cappings, okay. You have to see what is in between the capping, that is A1 to A4. Adopts a well defined beta turn. The dihedral angles of A2, A3 determine the type of beta turn. So is which out of these is the correct statement? As you know, Ramachandran plot is for the protein structures, finding out the secondary structure of protein. This was a previous method. Now this is not used so much in active research, but sometimes you get questions from this topic in CSIR. Okay, Malini says option 3. How about anyone else? You want to try? I hope by now you are well prepared for the exam. You should be able to answer these questions easily if you have had a decent preparation. And in case you have selected Unit 1 for your preparation, Biotechnica suggests that if you are well prepared with 7 to 8 units from the syllabus, then you can easily clear JRF in unreserved category, that is in general category. And if you have selected Unit 1 among your 7 to 8 units that you're going to prepare for, then you should be able to answer this question with a decent preparation. Yes, anyone else wants to try? Come on, no one wants to try? Okay, any are you people aware of Ramachandran plot? Do you know what is Ramachandran plot or not? Can you please confirm that? See, you have to interact with me, only then you will be able to understand what I'm telling. Let it not be a one-sided lecture. If you interact, then I can correct you if any place you are wrong. This is a very critical time where the more details you would know about the exam and how to be careful for solving part C questions, it will be more better for you. Okay, see, I'll explain this one. Ramachandran plot is based on the angles phi and psi. So, to understand what is phi and psi, you should go deep into the protein structure, which is having a peptide plane. As you can see, this is CONH bond, which is called as the peptide plane. And if you talk about an amino acid, this is one amino acid with its C alpha group, hydrogen R group, carboxylic group, and amino group. I hope you know the structure of amino acids. And here, between two amino acids, you have the peptide bond formed. So this would be one amino acid, this would be another amino acid. Between these, a peptide bond is formed, and this is what has formed a peptide plane. Okay. Peptide planes are planar structures, and that is why they can't have freedom of rotation. However, the neighboring structures, they have freedom of rotation. So this is what will be called as the peptide plane, right? Now, however, this cannot undergo any rotation, but these single bonds over here, the peptide bond can't undergo rotation because this double bond will shift here, and it will form a semi double bond, okay, partial double bond. And double bond doesn't have freedom of rotation. So for this reason, you have the phi and psi angles, which are beside the C alpha. The NC alpha bond is called as phi. It means that if this can rotate 360-degree, these different peptide planes would be taking different geometric orientation. So these are called as dihedral angles. Okay, and if you are forming a particular secondary structure, let's say alpha helix or beta sheet, parallel, anti-parallel, or any kind of beta turns, right, or any other type of helix, then they would have different phi and psi. These are the standard values. Okay, and you are supposed to know the standard values. Now, what is the question saying in statement B? That if you have the phi psi angles both in the negative range, negative degrees. Okay, now you will see that your alpha helix, both phi psi has negative values. Then right handed 310 helix also has negative values, and this also has negative values. Okay, both phi and psi is negative. Now generally, if you have a standard Ramachandran plot, 310 and pi helix are rare forms of helices in protein structure. Alpha helix is pretty common, and that falls in this quadrant. See, this is the graph that we draw. On the y axis, we take psi, and on the x axis, we would take phi. Okay, so there would be certain allowed angles of this phi and psi, because if you have two non bonded atoms next to each other, so there can be restricted movement. This plane cannot come too close, otherwise, there would be steric hindrance with this R, right? That's why this alpha helix, as you can see, it is coming in this quadrant of the Ramachandran plot. Okay, now what is statement B saying? That all the residues have phi and psi negative values. Okay, but it is also saying that the strand will be the motif will be protein structure will be helix turn strand. How is that possible? If you are getting the secondary structure as helix turn strand, then Ramachandran plot also should reflect phi psi values of that secondary structure. But since it is not matching, that is why B is not correct. Okay. Glycine is a very small molecule, and that is why it can allow rotation, freedom of rotation of phi psi. Because for glycine, this R group is just a H. That is why if you have R group from the C alpha, then there can be less hindrance. That is why phi and psi will be more for glycine. And whatever this shaded areas, these are the areas of allowed angles of phi psi. And that is why in glycine you would have many shaded areas which would have the phi psi angles in the allowed region. That's why it is said glycine has the largest area. This is a correct statement. Okay, and a peptide, this one is adopting a well defined beta turn. So you have four amino acids which, yes, it takes beta turn only. Let me remind you, there are two different types, many types of beta turn. Here they are talking about type 1, type 2. So the difference is in the peptide bond orientation between the second and the third amino acid, and that is what makes them a different type of turn. Okay, so this is what it is said. The dihedral angle of A2 and A3 will determine the type of beta turn. So this is also correct statement. Allowed value of phi psi are not valid for short peptide. Why it won't be valid for short peptide? Okay, if even if it is a dipeptide, then even a dipeptide would have a peptide plane, and as long as you have peptide plane, you will have at least one phi and one psi. That is why C is correct. So your correct answer over here is option 3. I hope everyone understood that. Let's look at a question. So these are your standard values and the Ramachandran plots of certain kind of structures like antiparallel, collagen, right-handed twist, left-handed twist, and all that. Okay. Then a recent question, December 2023. Which of the following sugars will not reduce tolerance reagent? This is from Unit 1B, carbohydrates. You're supposed to know the structure and also the characteristics of carbohydrates. Try to answer this. So you have certain structures. You would know that the carbohydrates are categorized into reducing sugars and non-reducing sugars. So basically the question is asking which out of this will be a reducing sugar, right? Now if you know anything about carbohydrates, then I'm sure you would have come across the term anomeric carbon. Now what is anomeric carbon? When you have a carbohydrate, first of all, there would be some carbon chain. Let me take hexose sugar only because here also they have taken 6 carbon sugar, and they can either exist in linear form. First of all, there would be a functional group, so here it is CHO only because all of these are pure pyranose rings, and which means they are from the aldehyde category. These are aldo sugars, okay. And then you will have CH2OH, and then in this OH, AH, all that will be there, okay. Like that, these are carbohydrates. So the first carbon, which is having the functional group, it is not anomeric in this case, okay. But what happens is that this OH group, it would make a attack on this carbon, because of which it forms a cyclic form, and generally carbohydrates will be present in the cyclic form. Now this sixth carbon, so this will be in the form of a ring. This would be coming in the ring. So this would be the 5 carbon, and one oxygen would be there, and then this would be the carbon, and then other carbons 1, 2, 3, 4. So here you will have 1, 2, 3, and 4. Okay, this would be the fifth one, and the sixth one, it will be outside the ring, CH2OH. Okay, so all of these groups, what you are seeing, you will see one carbon which is outside the ring. You can see this is the main sugar, and this is outside the ring, means this carbon. Okay, and with the carbon you have this OH group, which is a CH2OH. And to all of this individual carbon you have HOH, HOH, okay, HOH, HOH, right? So these groups, they need to have this anomeric carbon. This was not chiral initially, but later it became chiral. Now if this carbon is free, then this can actually form another glycosidic linkage, means it can form a glycosidic linkage, okay. So in case it is forming a glycosidic linkage, then the anomeric carbon will not be free. But if it is not from the other end, glycosidic linkage is formed. For example, we know the AHOH present towards the fourth carbon or towards sometimes sixth carbon. So that would form a glycosidic bond. In that case, the anomeric carbon would be free. Okay, so if anomeric carbon is free, then this carbon will be able to reduce another molecule. Okay, so generally we will be using some reagents for that, where some copper agents would be there, copper ions, and this copper ions from copper 3 plus, it would be reduced to copper 2 plus, where electrons will be gained, isn't it? That would show up a color change in the reagent, and that would mark the presence of the reducing sugar. This we generally use in estimation of reducing sugar, okay. So that is why, yes, very good, Malini, that's the right answer. So we have the option 1, 2, 3. They are disaccharides with 1,4 glycosidic linkage, and the hemiacetal of the second ring is not involved in the glycosidic bond, hence the reducing end is free and will reduce the tolerance reagent. Okay, so it is asking which will not reduce. So all of this you can see, since the sixth group is here, here also sixth group is on top. Okay, so that means this would be the anomeric carbon. Obviously you have to see the orientation. This is the sixth carbon, this is oxygen, and in all of them you can see the C1 atom of aldose sugar in the ring structure, it is free since it is the aldose group. But here the anomeric carbon is this one. How do you know? Because this sugar is in the flipped orientation. You can see this is the C6 carbon, okay, and this is the oxygen. So this one is C1, and C1 here is involved in a glycosidic bond. That is why its anomeric carbon is not free. If you treat them with reducing agent, this will not be able to reduce the other one. Okay, did you people understand? Are you people following or not? So actually when the ring like structure is formed, okay, from a linear structure you form ring like structure, this is called as a hemiacetal group. Okay, it is formed by the reaction of an aldehyde and an alcohol. Aldehyde from the first carbon of aldose, and aldehyde from the alcohol from the fifth carbon of the sugar. Okay, so both of them together react to form hemiacetal, and this can even reduce one more alcohol. It can join with another alcohol also, hemiacetal, to form an acetal group. But for that it needs to be free, then only it can reduce. So correct answer is option 4. Okay. The next question from December 2019. The following observations are made on a 30 residue polypeptide. This is again from protein structure. Unordered structure is observed in water, but a helical conformation is observed in medium of low dielectric constant. And they have given some observations. These are, you don't have to tell which is correct, incorrect here. All these are correct. Peptide is resistant to degradation by protease. Red blood cells are lysed by the peptide 30 residue, what they're asking. And beta mercaptoethanol has no effect on peptide structure. So, based on these observations, you have to tell which out of these options is correct. Yes, come on. I want a response from everyone, whoever is attending. Try to answer this. Don't be scared of whether it would be right or wrong. Don't want to try? Okay. So see here, um, generally we know that in proteins we have L amino acids, isn't it? And that is what have been coming through the evolution. Okay, no problem, Malini. So that's a good answer, because if you any question you're not sure, you also have the option of skipping, right? If you want, you can skip it. If you're not sure, you have been given the choice also. Okay. Okay, Nidambari also says 2. Okay, let's see. So generally we know that the amino acids present in proteins is generally L amino acid, isn't it? And our proteins, enzymes, they all will be habituated with this L amino acid. So what happens is, first of all, let's look at one by one. Unordered structure is observed in water. But what do we know? In proteins, in cellular environment, we would be having the folding of a protein chain in such a way that the polar groups will be present towards the outside, because in aqueous environment, cellular environment means it would be water would be everywhere, isn't it? And the hydrophobic groups, they will be hiding towards the core. Hydrophobic groups, they would be towards the core of the protein, isn't it? So for polar amino acid means these are going to be in ordered conformation in water, isn't it? So it is observed in water, but a helical conformation is observed in medium of low dielectric constant. Now water is a environment which has high dielectric constant. Okay, dielectric constant value, it has 80. What is this dielectric constant? It is a measure of the polarity. Water is a polar molecule. We know that water, double bond hydrogen, hydrogen, right? So we have oxygen more delta negative. That is why the hydrogen side would be delta positive, whichever is hydrogen side. Okay, so water is a polar molecule. That is why it has high dielectric constant. And when the protein is present in high dielectric constant, the protein folds properly. That is why it gives rise to a ordered structure. This I'm talking for regular proteins in cellular environment. Now, regular proteins, what did I say? They have L amino acids, isn't it? So first thing is that unordered structure is observed in water. Okay, that means in water it is not forming a particular kind of order what we were seeing before. What does that mean? That there might be um, different kind of amino acid present here, right? But a helical conformation is observed in medium of low dielectric constant. But when you are taking a non-polar environment, so non-polar environment, low dielectric constant means a hydrophobic environment, a non-polar environment, the protein is showing a helical structure. Okay, so helical structure means some groups it would be hiding inside, some groups it would be hiding outside, it would be exposing outside, which means that this protein would definitely have some polar group or non-polar group. If you don't have the two kinds of group, polar and non-polar both, then it won't be able to give an ordered structure in any environment, be it polar environment, be it non-polar environment. In any environment it wouldn't be able to give such a structure, stable structure. But in hydrophobic environment it is able to give a stable structure, means this protein would have both polar and non-polar groups. That means this protein has to be amphipathic. Okay, definitely it would be amphipathic. Amphipathic means something which has both types of groups. But it is said peptide is resistant to degradation by protease. Now here, generally, your L amino acids would be sensitive to the proteases, because all the enzymes they would be working on L amino acids as is present in the cell generally and normally. So if they are resistant, means their orientation should be something different. And the biggest thing is beta mercaptoethanol has no effect on peptide structure. We know that some protein parts they would be stabilized by disulfide linkages, and disulfide linkages can be broken by the effect of beta mercaptoethanol, which is a reducing agent. Okay, so only if the peptide has a different kind of orientation, so it would be a mirror image, then only the protein structure would be also different, in which the beta mercaptoethanol will not be able to break this disulfide groups, means disulfide group won't be there, because if disulfide group is there, then beta mercaptoethanol would be able to break it. Okay, so that means this peptide stabilization is through something else. That is why correct option is answer 1. Okay, peptide bonds formed by D amino acids are more resistant, and beta mercaptoethanol breaks disulfide bond. If there is no effect, that means it does not contain any cysteine residues. Okay, and unordered means they lack a fixed 3D structure, and they would contain high proportion of polar and charged amino acids. Okay, so that is why they are not able to form a particular structure. And unordered in observed in water, but a helical is observed in media of low dielectric constant, means they are amphipathic. Let me remind you that in case your preparation is not so good this time, or you are little bit shaky about the questions that we are discussing, then you can also aim for the December exam, December 2024, even the June 2025 also next year. In any case, students who are here in your final year of MSc, or even in your first year of MSc also, or your BTech exam also, then you can think of joining this batch, because you will be learning all the details from the very scratch, from all the basics of all the 13 units. Entire syllabus will be taught to you, and everything in detail. Okay, so this batch, regular batch, it starts 1st July 2024, classes will start. And we have very limited seats left actually. because already students have started enrolling, and in fact it's about to close anytime soon now. So that's why if you want to enroll, you have to take the decision fast. Call us on our toll free number, which is given here, or the office number, or you can even visit the stores to see what all you're getting along with that. Okay, so you would also get, since it is for December 2024, Biotechnica wants you to have a full round of preparation, and that's why everything like chat support, test series, and even general aptitude practice papers, the revision series, mind map, internship, study material, even concept table, concept wheel, all this will be there. And most importantly, there is double success guarantee also in case you're not able to clear on December 2024 after the coaching, then you can also keep it for June 2025 with a nominal. Okay, so you can contact on the number given, Graham, to know about that. Now, next is June 2021 question. How many hydrogen bonds involving the CO and NH can be observed in an alpha helix consisting of 15 amino acid residues? Anyone wants to try this? How many hydrogen bonds will be there? See, let's say there is a 15 amino acid residue which wants to take up an alpha helix. Okay, so from this alpha helix there would be some hydrogen bonds which will be stabilizing this helix, isn't it? So how many hydrogen bonds will be formed over here? That is a question. So for this you should know what is the pattern of hydrogen bond first of all. Yes, Malini, very good, the answer is right. So in an alpha helix, the hydrogen bond forms between CO of ith residue to NH of I plus fourth residue. So here, yes, Malini, that is right. We have the CO group. This is the first amino acid. This is the first amino acid. Then this is the second amino acid. You can see between the first and second, you have the peptide bond formed. Okay. Now, like this, our C alphas, they are marked as, so this is C alpha 1, this is C alpha 2, so this is I plus 1. Why is the first residue is called as residue I, or you can also call it residue N? Because this won't be the first residue in the polypeptide chain. It's not that from where the polypeptide chain starts, from there only helix starts. No. So helix would be like first few peptides will be just like that, then the helix can be, then the next stretch of protein will form the helix. So like that, we don't know whether it's the first or the 10th or the 21st amino acid of the protein. That is why we mark it ith residue or nth residue. The next amino acid would be I plus 1 or n plus 1. Like that, it would continue. Okay. Then, what is the hydrogen bond here? NH of I2, CO of I2, NH of I plus fourth residue. Now, since this is residue 1, CO of I. So, this is the CO of I, and I plus 4 is this residue. Okay, so that means this is the C alpha, and always we will label our protein from N to C terminal, right? So if we, this is the I plus fourth residue, this is the NH of I plus 4, this is the CO of I plus 4. So if we have to take NH of I plus 4 means it is this group. Okay, so as you can see, these two groups are joined with a hydrogen bonding, correct? Now what would be the next hydrogen bonding? You have to take the next CO group, which means the next hydrogen bonding would be CO of I plus 1. So this is I plus 1, this is a CO, and NH of I plus 5. So this is I plus 5, this is the NH of I plus 5. These two will join together. So like this, the hydrogen bonds will continue until 15th residue. So how many hydrogen bonds can be formed? So if you take a 15 amino acid residue, let's consider first residue is I8 and 15th residue would be I plus 14. Because I you have taken as 1, so I plus 14 will be 15th. Here are all your residues. So either you can draw one by one the residues, but better is like you can write like this. Okay, so 10th hydrogen bond would be CO of the 11th hydrogen bond will be CO of I plus 10. So where is I plus 10? This is I plus 10, this is the CO of I plus 10, and NH of I plus 14. So this is I plus 14 till NH. So this will be the hydrogen bond. Now for the next hydrogen bond, you have started from residue 1, how I showed. So like that, this is the 11th hydrogen bond. Now if you have to form the next hydrogen bond, that is from this CO group, then you would need four amino acids in between that, because our bonding is going I to I plus 4, which means we need four amino acids in between. But if this has to bond with the next one, then you would need a CO of NH group of I plus 15. But there is no I plus 15, because this is only till the protein structure. So there is no I plus 15. Okay, so that's why this CO cannot form a bond. This also cannot form, this also cannot form, this also cannot form. So the last four CO will not be able to bind. Okay, they would have only hydrogen bond with the NH groups, not with the CO groups. Similarly, the first four NH bonds will not be able to form a bond. They can only form a bond with the CO groups, because CO are to NH group. That is why the first four will have only half of hydrogen bond. Okay, half of hydrogen bond. Last four also will be having half of hydrogen bond, and in between they will be having one hydrogen bond. So like that. So best trick is that you have to subtract minus 4 from whatever is given. So for example, here they have given 15 amino acid residue. That means 1 to 4 it will be half. So half into 2, half into 4 is 2. 4 halves is 2. Then fifth to then again 15th, 11th to 15th, sorry, 12 to 15 will be again 2, and 5 to 11 will be 7. Okay, so I said in between the amino acids they would have one hydrogen bond, whereas 1 to 4 and 12 to 15 they would have half, so 2 2. So 2 plus 2 plus 2 is 11. But you can directly subtract 4 from whatever is given for such kind of questions. So like that, answer would be 11. Okay, the next question is from June 2021 again. This is regarding a CD spectra in the far UV region. We know that these tell us about the secondary structures in the protein. It informs on the kind and secondary structures in protein. Near UV and tryptophan emission spectra will tell us about the tertiary structure. Okay, whereas the UV spectra, CD in UV will tell us about secondary structure. So here you have some spectra which is shown. This is for intrinsic fluorescence emission spectra of protein X. This is far UV CD spectra, and this is near UV CD spectra of protein X. And these are recorded under different conditions. So red, green, and black, they represent all different condition. So basically, you have the intrinsic fluorescence emission in pH 7. So the black color is for pH 7. pH 3 is the green color, and pH 7 is the red color in presence of guanidine hydrochloride. Okay, so we know guanidine hydrochloride is a chaotropic agent which denatures the protein. Okay, if we have students from our previous regular batch, they would know. You would be able to say this, Malini, right? So go, as we have studied in our denaturation chapter, guanidine hydrochloride is a denaturant, right? So it would be able to disrupt the tertiary structure of the proteins, even the secondary structure also. And similar effect even pH 3 would be having, because pH also acts as a denaturant, and they have shown different curves. So what does the question report? What does the experiment report? These different groups, even if you see in far UV spectra, far UV spectra also tells about the secondary structure, whereas the near UV spectra and near UV spectra and emission spectra, that is tryptophan emission spectra, this would tell about the tertiary structure. Okay, so in far UV CD spectra also you can see this is the black line. If you would know the secondary structures like alpha helix, beta sheet, and random coils and turns, uh, what does the spectral line look like in general far UV CD spectra, then you would be able to say that the structure is definitely disrupted in case of the red color, that is the presence of guanidine hydrochloride. Okay, as you can see, it is a flat structure. And even in pH 3 also you can see random coils. So anyways, out of this, what would be the experiment reporting? What do you think? Like whether protein is fully folded at pH 7, or protein is reduced at pH 7, or what happens, or there is extensive denaturation both at pH 3 and in 6 molar guanidine hydrochloride. Changes in fluorescence and near UV indicate increase in hydrodynamic radius at pH 3. So what is this? Yes. So option 1 is the right answer. Protein is fully folded at pH 7. As I just told you that pH 7, we can see here, so see Palakpreet and Malini, we have the, as I told you, we have the emission, the tryptophan emission. It is happening for this black line, which is at pH 7 in the zone, regular zone. Okay, and you can see that the means whatever zone you are having, as you are changing the pH. For pH 3, you can see the emission maxima is even more. Okay, and if you are using guanidine hydrochloride, then also your emission maxima increases, which means the protein is getting unfolded. Okay, as guanidine hydrochloride, it will be completely unfolded, and in pH 3 it has increased little bit. That is why we can say it goes into a molten globule state, where it is not completely randomly opened in a linear chain, but they would be having like partially some parts will be fixed together. Okay, yes, Anupam, that's the right answer. Very good. Okay, and even here also you can see that protein is showing the regular turns, but in guanidinium hydrochloride it is completely unfolded. That is why it is showing a full straightened out line. Okay, whereas you can see that the pH 3 green color and black color, they are more or less showing that the structure is retained, because far UV, it is about the secondary structure. So if a structure is in molten globule state, this is the difference in near UV and far UV. In tertiary structure, you would see the green color line is also showing a disrupted structure. Okay, it is only the black color is showing a proper tertiary structure, but you can see both the green and red color are showing flattened spectral line, which means in both of them these are unfolded. That means the molten globule does not have tertiary structures fully fixed, it has some secondary structures fixed. So that is why in far UV, the green color line, which is treated at pH 3, you are able to see some secondary structures which it is showing. Okay, so that's why one is the right answer. Did everyone follow this question? Yes, this type of questions also you would be getting in your CSIR. Like this is with reference to Unit 13, in combination with Unit 13. Okay, and while we were at this part, like hydrodynamic radius, I was one of the options was saying for native folded proteins it is this much Armstrong, and for highly denatured protein it will be less. Remember, hydrodynamic radius for native proteins will be high, but denatured proteins it will be less. Okay, so hence with denaturation, which is indicated by changes in fluorescence and near UV, it should mean that the hydrodynamic radius should decrease with the unfolding of the proteins. Okay, but since in statement 3 it is saying increase, that is why it would be wrong. Okay, okay, try this question. It is from December 2015. So this is a very simple question. Let me see how many can answer. This is a very good example. Like they have given unnecessary details over here. If you have just got exactly what they are asking, then you can very easily answer this question. But yes, to solve this question, you need a knowledge of amino acids, different type of amino acids. Anyone wants to try out? See, in part C, you can expect such kind of experimental based questions only, where so in classes also we discuss the basic concepts, and then only we will be discussing this kind of application based questions so that you're able to answer correctly. Okay. See, this also requires a little bit of unit 13 concept. These That's why try to select the units wisely, whichever units you're supposed to prepare. Why? Because if you're preparing unit 1, and or else let's say you're preparing unit 1, but you're not preparing unit 13, but that's not a good choice, because 1 and 13 has many related questions, and same with vice versa. If you're preparing unit 13, which is lot of questions based on biochemical basics like pi point, isoelectric point, or even the protein structure determination, so separation of proteins, so these all will require um, the knowledge of biochemical structure as well. Okay, so if you're leaving unit 1, that's not a good choice. That's why when you are starting with the Josh batch, if anyone is interested, we would be giving you some tips on how to select units, which units you should study together, right? Even you can look at the other videos of Biotechnica. Anyways, four single amino acid mutants to a protein. So basically, you have a monoclonal antibody, and the area with which it will be recognizing that area, it is made. Some mutations are done in the epitope region. Some mutations are done to it. Okay, and then each of these are expressed in E. coli. So each of these E. coli, they will be making these four proteins, and these proteins are run on an SDS page gel, and blotting was done, western blotting. So on the nitrocellulose membrane, you have the bands which came. So this was the load control on nitrocellulose membrane. You had the load control where where these, what to say, the different mutants, four single amino acid mutants, they were given. So this was the wild type epitope region. Okay, and these are the ABCD 4 mutants. Now you have treated this nitrocellulose membrane with monoclonal antibody, okay, the wild type antibody, and you saw with what it is binding. Obviously, if you have wild type monoclonal antibody, you can see that the protein is able to recognize the epitope properly, isn't it? You can see. That is why I hope blotting, you know the technique. Wherever its complementary molecule will be present, protein's complementary molecule, there the protein will go and bind. Okay, on the membrane itself it will bind, and then you use some tags to identify the antibody. Okay, so for its presence, that means here monoclonal antibody has bonded, means the epitope region, it is wild type, it has not changed. Okay, same in B also. If you now compare the amount of binding, you would see B also has same amount of binding, which means that the mutation, whatever single amino acid mutation B had, it did not change the nature of the amino acid, because of which it is not changing the overall folding of the epitope region, and that is why it has the same binding like that of wild type. Okay, but in A it is slightly reduced. Okay, and in C and D there is no binding at all, which means whatever amino acid mutations have happened in C and D, they led to the folding of the protein epitope region in such a way that the antibody is not able to recognize anymore. Okay, so here you need the mutations are given over here, valine to alanine. So clearly we want to check for C and D, right? Because in C and D we need a drastic change in the type of amino acid. So here you have C and D, and here you have C and D. Okay, so one is said valine to alanine. Now, valine alanine both are non-polar amino acids. So that is why if both are non-polar means if you change from valine to alanine, there won't be much of a difference in the protein structure, especially the folding part. We know in folding, non-polar amino acid are buried inside, polar amino acid are buried outside. So not much change. So that means 2 cannot be the answer. Okay. Similarly, here also C is due to valine to alanine. Even 4 cannot be the answer. So out of 3, even if you see here isoleucine to leucine, See, here also you need a lot of knowledge, not only the nature of amino acid, but also the single letter codes as well. So this is isoleucine to leucine. Again, these two are non-polar molecules. So none of this is the answer. Only option one is the answer. It is that easy because glycine to proline, it's a drastic change. Glycine is the smallest amino acid with a very simple polar group, which a very simple side chain that is just hydrogen, and proline is a very complex structure which has an imino ring. The side chain is connected with its own amino group. So, polypeptide chain with glycine, how it would fold, but a polypeptide chain with proline, it would won't have that kind of structure. Okay, so that's why option one. Now, B, I told you B shouldn't be much change because it is giving the same nature. So, valine to alanine makes sense because both are non-polar amino acids also. Okay, I hope you got that. This is the explanation which is given. Right, you can see from the recording later. Okay, next question from November 2020. This is regarding Anfinsen's experiment. In classical Anfinsen's protein folding experiment, good, Malini. In classical Anfinsen's protein folding experiment, the enzymatically active ribonuclease is treated with beta mercaptoethanol and 8 molar urea, following which the preparation was dialyzed to remove beta mercaptoethanol. Sample was completely oxidized in 8 molar urea. Traces amounts of beta mercaptoethanol were added, and 8 molar urea were added to the dialyzed sample. So, which of these would lead to full enzymatic activity of the ribonuclease enzyme? So generally, this days questions regarding the famous experiments can also come, or some historical perspective. Like one time there was a question regarding the X-ray diffraction pattern of bDNA and aDNA. bDNA, the one Rosalind Franklin gave the structure. So do not ignore this historical perspective or the famous experiments because from there also experiment and the questions will be coming. So answer here is A followed by C because if you know something about the Anfinsen experiment, he did with a native ribonuclease which is catalytically active. It has 4 disulfide bonds in its structure, and only if it is folded in this form with exact this disulfide bonds. See, total 8 cysteine residues are there, and 8 cysteine residues can join in 105 different ways, permutation combination, but there is only one way that the protein will be functional, and that is when these disulfide linkages would be binding to each other. Okay, now here the first of all, if you add beta mercaptoethanol and urea will get denatured. Okay, urea denatures, and beta mercaptoethanol also reduces, so ribonuclease A will denature, resulting in complete loss and unfolding of the protein. Here you can see that the protein has completely unfolded. Okay, the cysteine residues have unfolded from each other. Now you have added 8 molar urea, and along with that you have removed the beta mercaptoethanol, but still urea is present. So upon removing beta mercaptoethanol, which is a reducing agent, it cleaves the disulfide linkages. So you can see some disulfide linkages have are able to form, but because of urea they are still in the opened up structure, randomly formed disulfide bonds. Okay, because urea is not allowing it to come to a stable structure, so it is randomly forming any disulfide linkage. However, this protein will not be active. Okay, so if you remove BME first before urea, it will lead to only 1% recovery because random incorrect disulfide bridges will form. Okay, then if you remove the urea first, okay, and then allow the beta mercaptoethanol to be present in trace amount, then what happens is that upon removing urea, it would obviously become stable, and on top of that the but even if it becomes stable, there is a chance that incorrect disulfide bonds would have been found between them. But if that happens, if you put beta mercaptoethanol in trace amount, then it is possible that whichever incorrectly bonded disulfide bonds were there, those will be broken and the correct ones will form. So removing urea first and allowing protein to refold before disulfide bonds were restored with oxidizing agent, it will lead to recovery of 90% of activity. Okay, and that's why A followed by C will be the correct. You first have to dialyze to remove both beta mercaptoethanol 8 molar urea, and later you just add some trace amount so that whatever incorrectly disulfide bonds have been formed, they can be opened up and they can form in the correct way. Later, anyways, you can remove the beta mercaptoethanol. This is a similar type of question regarding protein folding. I will not discuss this. Let us discuss some question from enzyme kinetics because of lack of time. So this is also from December. This is from November 2020 exam. The Hill equation and its plots describe the following enzyme kinetic behavior. One is saturation kinetics, cooperative kinetics, or the this one the equation, which out of this equation is correct, and whether it describes saturation kinetics and or cooperative kinetics. So basically one out of A and B should be correct. Hill equation describes what, and one out of C and D should be correct. So that's why you have two options, correct options, that you have to choose from here. Yes, anyone wants to answer this? So are you aware of the Hill equation? What is it for? It gives a straight line graph equation for the binding of protein and ligand. So this is also applicable for enzymes as well, because enzymes are also proteins and ligands are the substrates. Okay. So what happens over here is we have the Hill equation. You should know how we get it. Um, yeah. So we have the Hill equation, which is a measure of cooperativity. Always remember whether it is cooperative or not. We have studied in hemoglobin also. Hemoglobin, myoglobin. Myoglobin is non-cooperative. Hemoglobin is cooperative. Okay. So if we have the enzyme, and let's say the enzyme has n number of binding sites in, so substrates will be like n substrates, means if the enzyme has, let's say, 4 binding sites, so n substrates, 4 substrates will be required so that the enzyme forms a complex with 4 substrate molecules. Okay. Then you have the dissociation constant. So dissociation constant, we know Kd, it would be the reactants divided by the products. Okay, if you are selecting K, then it would be other way around. So here we define something called degree of saturation, which we represent as Y, or even in some proteins we can also represent it with theta. Okay. Now, this is given by the total number of binding sites. So, total number of binding sites means the total number of enzymes. And on numerator, how much are bonded with the substrate. So, how much are bonded with substrate means the concentration of the ESN. That means how much is in complex with the substrate. And E0 represents the total number of binding sites. Okay. So to bring it into a straight line, we know that this equation, when you have the regular Michaelis-Menten equation, then it represents a hyperbola like this. Okay. But we want to get a straight line graph equation. So what would we do? We would make some changes. We would make 1 minus I, 1 minus y. So then you can reverse, then you can keep it in log form. Log of y 1 minus y. Anyways, I'm not going into the derivation. This is the equation that you would get. Now, in case of enzymes, this y would be V0. That means what is the degree of saturation. And 1 would be the maximum saturation. See, the maximum value of binding can be when all of binding sites are occupied. Okay, so 1 is the maximum value of theta. Okay, so in which 100% of the sites are occupied. And this same thing happens with enzymes for Vmax. Vmax is the velocity where all the enzyme sites are occupied with substrate. Then only in saturating substrate concentration you get the maximum velocity. So you would have the Vi log of Vi or V0 divided by Vmax minus Vi is equal to n log substrate concentration minus log of the dissociation constant. Okay, so we know that this log this falls in the form of y is equal to mx plus c. This is the equation, and since you are getting this y axis, we have to take it in this y axis whatever we have mentioned here, and in the x axis it would be log of substrate concentration. Then this it would represent the what to say y-intercept. That means where the graph line is crossing the y-axis, that is y-intercept. So since here it is crossing in minus, that's why it will be minus log k. This point will be equal to log k. So from the graph you can also find out the dissociation constant, and m would be the slope. So how much inclined it is. Let's say you can have a slope like this, then the slope is less. You can have a slope like this, that means the slope is more inclined to y-axis. Okay, remember, if slope is more than 1, that means the protein is cooperative or the enzyme is cooperative. Okay, if it is equal to 1 means it is non-cooperative. So log L values near the region of the curve that crosses x-axis approximate a straight line with a slope of 2.8, which implies that there is maximum cooperative in the middle of the binding curve. So when n is equal to 1, as it would be with myoglobin, the number of other oxygen bound to other site, irrespective, they will bind with same affinity. Okay, but in hemoglobin example, what happens? You have 4 binding site. So when one ligand binds, okay, the next ligand binding is easier, the next ligand binding is even easier, and the last ligand binding is very easier. So generally their plots will be more than one, right? So that's why here answer will be cooperative kinetics, and this is what we have log, this part is right, but it would be cooperative kinetics. Okay, Malini, got it? So this was about the Josh batch. I would like to remind you that there are very limited seats available, so you should hurry up. Okay. And then let's solve this one question from June 2016. This is from carbohydrate metabolism. But however, this question is also important, this type of question, because it is based on electron transport chain inhibitors. Okay. So here we know that electron transport chain, it has several components, complexes, and different inhibitors will inhibit at different complexes. Like rotenone will inhibit complex 1, antimycin would inhibit complex 3, sodium azide would inhibit the complex 4, and even cytochrome will also inhibit carbon monoxide. Carbon monoxide, they all will be cyanide, not cytochrome, cyanide, carbon monoxide, they will be inhibiting the complex 4. But whatever is the inhibitor, if one of the inhibitor you are using, then the entire electron transport chain will stop, because electron transport chain includes all of these. Okay, in that case the electrons will not be given to oxygen, which is terminal electron acceptor, and in that case there will be no proton gradient, proton motive force formed, and no ATP synthesis will be there. So oxygen will not get used up. If electrons are not given to oxygen, it will not utilize H plus, it will not form water, which means oxygen concentration will not decrease. Okay, it will remain same. Now, there are some inhibitors which are called as oligomycin. Those are called as, those will inhibit the ATP synthase. We know that once the proton motive force H plus ions are transported, they would try to come back through the membrane through the F0 F1 particle. Okay, so when they come through the F0 F1 particle, ATP is synthesized. But for this ATP synthesis, we want the proton gradient to form. But okay, now, generally, these two processes are coupled together, the electron transport chain and the F0 F1 particle ATP synthesis. But if they are, this ATP synthesis is blocked, okay, if this ATP synthesis is blocked by using oligomycin, then what happens is this ATP synthesis will not be able to happen, okay, and slowly these H plus ions which were accumulating, they will not be able to come through the other side, so there would reach a saturation. Okay, any more H plus cannot be sent, and that's why eventually electron transport chain will also slow down. But there is a molecule which is called as dinitrophenol. it is called as an uncoupler. So what does it do is, it will remove this H plus ions, okay, it would remove the H plus ions, and because of which, even if F0 F1 particle is not working, but still electron transport chain will keep on taking place, because even though H plus gradients are transported to the other side, they would be constantly removed by the DNP, which is the uncoupler. That's why even if ATP synthesis is not happening, electron transport chain will still happen with this uncoupler, because they have because this DNP has uncoupled the two processes. Okay, so this is a question in a mitochondrial respiration experiment. A researcher has observed the following profile. It is asking for oxygen consumption. So oxygen consumption means you just have to say whether the electron transport chain is happening or not. Okay, so oxygen consumption is not happening here, and then after you add something, oxygen consumption increases. This is only possible when electron transport chain is increasing. Okay, then this slows down, electron transport chain slows down. You can see it has not stopped completely, otherwise it would have become constant. It has slowed down very much. Then again, if you had added something, then again it has increased. So what could be these things? Obviously, the first increase can be when you have added any substrate. So here succinate can act as a substrate. Okay, that's all. And ADP and PI can also act as a substrate. Okay, not substrate but precursors for ATP synthesis. So one can be either A or E. Okay, it can be either A or E. Then after adding 2, you can see ATP synthesis has slowed down. So this is only possible when you add oligomycin, because if you add cyanide, then it would completely stop. So that is why 2 cannot be cyanide, and 2 cannot be substrates also, because otherwise it would have increased more. So it can be C, and after adding 3, you can see again it has resumed. Now don't forget, it is in the same test tube. So 2 is already present. In the presence of oligomycin, you are adding 3. Okay. So 3 cannot be a substrate, because you have to think of the oligomycin being present. So something, it should disrupt the proton gradient. Then only it can keep on taking place. So this can be only dinitrophenol. So this can be B. So 2 being C, and 3 being B, and 1, in that case they have taken A. So 4 is the right answer here. Okay. So that's all from my end. Thank you everyone.